We might be right on the edge of a major breakthrough in the fight against Alzheimer's disease. There's this new drug that's showing incredible promise. But for a while, scientists weren't totally sure why it worked so well. So today, we're going to break down the discovery that finally solved that puzzle.
So we know this new therapy can clear out the toxic gunk that builds up in the brain with Alzheimer's. But the huge question scientists were asking was, how, right? What is the secret sauce here? Okay, let's get into it.
All right, let's start with the drug at the very center of this story. It's called Lecanemab. It really represents a new hope for treating Alzheimer's, but it also handed scientists a really fascinating puzzle to solve.
So get this. In clinical trials, Lecanemab slowed cognitive decline by 27%. It's what's called an antibody therapy, and we knew it worked by removing these harmful things called amyloid plaques from the brain. But for the longest time, the exact way it did that, the precise instructions it was giving the body, that was a complete mystery.
Okay, so to solve this puzzle, we first have to meet the key players inside our brains. I want you to imagine the brain as this bustling city. And to keep that city running smoothly, you need a very, very specialized cleanup crew.
The whole problem really starts with these amyloid plaques. They're basically toxic proteins that clump together. Think of them like garbage that just piles up all over the city, getting in the way and disrupting how brain cells talk to each other.
Now, the brain actually has its own built-in cleanup crew. These cells are called microglia. There's the brain's immune system, and their job is literally to clear out waste and keep things tidy. But for reasons we're still trying to figure out, they have a really hard time dealing with all that amyloid trash by themselves.
And this is exactly where lecanemab comes into play. You can think of it like a new supervisor showing up at the job site. It doesn't clear the trash itself. No, instead, it carries a very specific set of instructions to help the microglia, our cleanup crew, to finally target and remove those amyloid plaques effectively.
So, here's the crucial part. It's all about how this supervisor communicates. The antibody has a special part called the Fc fragment. Just think of it like it's walkie-talkie. With a working walkie-talkie, it can call up the microglia cleanup crew and tell them, hey, get over here and start clearing these plaques. But what if that walkie-talkie was broken? Well, scientists created a modified version where this Fc fragment was basically silenced. It could still find the amyloid trash and stick to it, but it couldn't talk to the cleanup crew. So this whole broken walkie-talkie idea, it became the key to a really clever scientific test. By comparing the drug with a working radio to one without, researchers could finally figure out exactly what was going on.
The researchers basically set up 3 different experiments. First, they gave the normal drug to mice with Alzheimer's-like plaques and a healthy cleanup crew. Simple enough. Second, they gave that modified drug, the one with the broken walkie-talkie, to the same kind of mice. And then, for the most critical test, they gave the normal drug to mice that had the plaques, but they had their microglia, their cleanup crew, completely removed.
And let me tell you, the results from this test were just stunningly clear. It provided the kind of visual proof that scientists live for. Let's take a look.
Okay, what you're seeing here is just a night and day difference. It's incredible. On the left and on the right, those are the brains that got either a control antibody or the modified one. They are just full of those little white spots. That's all the amyloid trash. But then, look at the brain right there in the center, the one that got the fully functional lecanemab. It is so much clearer. The cleanup was a huge success.
And this chart just hammers the point home with cold, hard data. It shows the percentage of the brain covered in plaques. You see the bars for the control group and the group that got the antibody with the broken communicator? They're way up there. But the bar for the standard lecanemab group is tiny. This confirms it. The drug only works when it can communicate.
But this is the final piece of the puzzle. This quote from the research paper says it all. In the mice that had no microglia, no cleanup crew at all, lecanemab did absolutely nothing. The plaques just stayed there. This proves, without a shadow of a doubt, that the drug is totally powerless on its own. It needs the microglia.
And this image from under the microscope gives us an even closer look. You can see the modified antibody, that's the magenta color, and it has successfully found and latched onto the green amyloid plaque. So it's sitting right on the trash, but because its communicator, that Fc fragment, is broken, it just sits there. It can't call in the cleanup crew to come and haul the trash away. So mystery number 1 is solved. Lecanemab works by using the microglia. But what the researchers discovered next was, honestly, even more amazing. It turns out the drug doesn't just give them a one-time order, it fundamentally rewrites their entire cleanup operating system.
What the research showed is that lecanemab is basically like a software update for the brain's immune cells. It actually triggers a complete genetic reprogramming, switching on this whole set of genes that turns the microglia into these highly efficient, plaque-destroying machines.
So this new genetic program supercharges a few key things. First, it enhances what's called phagocytosis. That's just the fancy scientific word for the cell's ability to literally eat the plaques. Then, it boosts their lysosomal activity, which is like upgrading their internal garbage disposal system to break that trash down better. And finally, it improves how they communicate with the rest of the body's immune system, to call for backup if they need it. And inside this whole new genetic program, researchers found one gene that seems to be the master switch, SPP1, which is also called osteopun. The drug makes the expression of this gene go through the roof in the microglia near the plaques. And in follow-up experiments, they found that this protein alone can help microglia clear away that amyloid. You see, this discovery is so much bigger than just one drug. It gives us a potential blueprint for the future, a whole new way of thinking about how we can use our own immune systems to treat diseases.
So, if there's one thing to take away from all this, it's this. The drug's success comes from a really powerful strategy. Instead of being a weapon that attacks the disease head-on, it's more like a coach. It retrains our body's own immune system to do the job it was always meant to do, but way, way better. It harnesses and empowers our own natural defenses.
And that really leaves us with a fascinating final question, doesn't it? If we can now essentially write new software for our immune cells to fight Alzheimer's, what's next? What other conditions, from different types of cancer to autoimmune disorders, could we start tackling by reprogramming our body's own defenders? The possibilities are truly exciting to think about.